is a brief review on DNA, RNA, and DNA replication. DNA is made of sugar, phosphate, and nitrogen base. This is the nucleotide that we see for DNA. It's DNA because it's made of deoxyribose, and we see that we have T's. It's found in the nucleus, and it carries the genetic code, and that code then codes for proteins. Base pairing rules: A always pairs with T, and C and G always pair together. So this is a good picture. The little dots show you the hydrogen bonds. The blue, dark blue, is the deoxyribose, the light blue is the phosphate, and then in between are our bases. And you can see red and green are always together, and purple and yellow are always together. RNA is made up of the sugar ribose, phosphate, and nitrogen base, and there is a U now instead of a T. There are no T's in RNA. RNA is found in the nucleus where it is made. It travels through the cytoplasm to the ribosomes. It carries the code for proteins from the nucleus, and it also carries amino acids. We'll look at that later on. We have the RNA here. It has uracil instead of thymine, and we can see it's single-stranded, unlike DNA. It is not double-stranded. It has a backbone just the same, but it has these U's instead of T's. DNA replication is the copying process of DNA. It copies before a cell is going to divide, and it shows semi-conservative replication, which means each new molecule retains one original strand and has one new strand. Enzymes, helicase, primase, and DNA polymerase all help for this process to occur. And DNA replication we can see here. The enzymes come in, they split apart the original DNA, the new strands are being added here and here, and this will happen all the way down until all the DNA has been copied.